Hello, everyone. Thank you for joining us for today's webinar, Mobilizing Science, Partnership, and Capability to Develop a Gene Therapy in Just 14 Months. I'm Heather Masaccio, your host from Cell and Gene and Life Science Connect. This webinar shares the remarkable story of how one family, propelled by urgent need and guided by gene therapy pioneers, partnered with Andelin Biosciences experts to develop a treatment for their daughter's ultra-rare neurodegenerative condition. More on that in just a moment, but first, I need to give you just a bit of housekeeping. Now, if you have a question for our speakers or wish to share any thoughts of your own, please submit it through the Q&A box on your screen. Our speakers will be happy to address your questions at the conclusion of the presentation during that Q&A session. If you encounter any problems with the stream today, please try refreshing your browser first. If your problem persists, go ahead and use that Q&A tool to submit a question, and we'll help get you back on track. This event will be available on demand, normally within about 24 hours. You can just use that same link that you used today. Now, it is my pleasure to introduce our presenters. First, we have Matt Nyloff, Chief Commercial Officer of Andelin Biosciences, and Catherine Mayer, PhD, Chief Scientific Officer and Head of R&D, Alcyon Therapeutics, and Michelle Kruger, Ellie's mom, mother to Ellie and co-founder of Ellie's Team Foundation. I'm going to go ahead and I'm going to turn it over to Matt to get us started. Matt?
Introduction and welcome everyone. Thank you for joining our webinar. Today we will walk through rare and ultra-rare disease and how to progress a gene therapy and address all of the challenges associated with progressing a therapy with speed. There are varying definitions of rare disease and ultra-rare disease, but in general, rare disease affects fewer than 200,000 people in the US alone. Ultra-rare disease may affect fewer than 100 people globally. And the impact is pretty vast globally. There are 10,000 known rare diseases, and this means over 300 million people globally are affected. Today, what you will learn is an understanding of how patient-led initiatives and organization of experts and partnership will enable driving forward therapy. You'll also learn about critical enablers to speed up a progression of a gene therapy program, and this is accomplished through parallel execution of scopes of work, digital modeling of the brain in this case study, and a well-characterized platform process, and also making bold decisions. We'll also explore a replicable framework for rare disease communities seeking how to move from diagnosis to treatment faster, and will include insights around regulatory, clinical, and manufacturing pathways. I will turn it over now to Michelle.
Thanks, Matt. Good morning, everyone. Thank you for your time, and I'm honored to be here and to share a little bit about our story. So first, I'm going to just start at the beginning. Ellie is our third child. She was born completely normal, and at about 5 months of age, in November of 2023, we noticed something was wrong. While she seemed somewhat delayed on milestones, as our third child, I wasn't as concerned about that, as I knew every child develops differently, and I imagined that it was just taking her a little bit longer. But what I couldn't ignore was there was this slight flicker in her eyes that I kept seeing. It started to get worse, so we went to our pediatrician, who ended up sending us to the emergency room. And it was the night before Christmas, or excuse me, the night before Thanksgiving. And she was really concerned by what she saw. I remember thinking, why do we have to go to the emergency room? This is probably just something silly. Can't we deal with it after the Thanksgiving holiday? But we listened to her and we went. We spent a long night in the ER doing every single test imaginable. We left eventually at about 4 in the morning, and we were sent home with no answers, and we were instructed to just await a call from the on-call neurologist the next day. The next day was Thanksgiving, and we spoke to Dr. Zachary Grinspan, who thought she might be experiencing seizures. I remember being in such shock, thinking as a mother to 3, that this was going to be something minor. Seizures was a big, scary word, and something that really put me kind of into shock. I remember Dr. Greenspan said the only way that we would know was to come back to the emergency room, get admitted to the hospital, and do an overnight EEG. So on Thanksgiving, we did that. And very shortly after being hooked up to the EEG leads, we were officially told that she was experiencing infantile spasms and focal seizures. I asked Dr. Greenspan what could be causing the seizures. And he told us, based on his experience, it would have been one of three things. One, just random, medications would resolve it, and she would go on to live a normal, healthy life. Two, some sort of structural issue with the brain, like cancer or a tumor, something like that. Or number three, a genetic disease. We immediately started praying and hoping for number one, that this was just a random situation that would resolve with medications and be a short part of her life. After a few months of aggressively trying to treat her seizures and not seeing any response, we accepted that we were most likely looking at something more serious. I remember praying and praying that the reason was number two, a brain tumor or cancer. I know as a parent that sounds crazy. Who prays that their child has cancer? But I knew in my gut that cancer and brain tumors were well studied, well understood, and well funded. I remember thinking that brain surgery would be better than the unfortunate reality of a rare genetic disease. So for weeks I prayed while we waited for answers. However, one day in February of 2025, we received the call from Dr. Greenspan indicating we had an answer. Ellie had a mutation in the IRF2BPL gene, causing a disease called netimus. After her diagnosis, we were, of course, in shock, but we set out to learn everything we could about IRF2BPL and netimus. It didn't take long to read the few papers on the disease and to talk to all the key researchers who knew anything about the gene. The gene itself was only discovered in 2018, and there are only approximately 150 kids in the world with the disease. Each child is very different, but many of the children struggle with seizures, abnormal movements, autism, and global developmental delays. We also learned that unfortunately, our disease is progressive and believed to be neurodegenerative. Some kids have learned to walk and to talk, only to regress later in life as the disease takes hold. I also remember sitting in the hospital asking Dr. Greenspan what was the treatment plan, only for him to tell me there were no available treatments or cures. He paused and slowly continued by saying, but gene therapy is becoming a reality. And if you want to set down that path, I will do everything I can to help you. I remember that moment so vividly, and the days that followed, where grief overtook us. When you receive news such as this, you can either let the sorrow take over or you can fight. And so we decided to fight. The first step was learning everything we possibly could about genetic diseases and the treatment pathways. I was introduced to an amazing mother of a friend who had already set down this path for her brave son. This mother spent 2+ hours with me walking through everything I needed to know about genetic diseases. She clearly articulated the importance of first understanding the disease mechanism and then immediately seeking the 4 paths of treatment: gene therapy, ASOs, drug repurposing, and gene editing. Coming out of this conversation, my husband and I decided to start Ellie's team, a faith-based foundation with a singular focus on translating medical research into treatment in record time. In a few short weeks, we built a team of incredible scientists and organizations who committed to helping us on our path to treatment. Our partners are listed here on this slide, and we would never have been able to accomplish what we did without every single one of them. mentioned, we were taught that there were four main paths forward. We decided to pursue all of them at the same time. Ellie's disease was progressing, and we knew we had to move quickly. We knew our best shot on goal was to take all treatment options as far as we could, and then eventually pick the best option when it became clear. One promising approach was small molecule repurposing, where we worked with a company called Unravel out of Boston that used Ellie's RNA data to predict potential drug candidates that could alter the disease progression. We down selected from this list to validate the lead drugs in a disease-induced tadpole model, which is still ongoing. We also engaged the JAX Labs Rare Disease Translational Center and worked with Kat Lutz to make a case for a humanized IRF2BPL mouse model. JAX saw the value and funded its development, which is still in process. Concurrently, we explored ASOs, meeting with leading developers at the Enlorum Foundation, the lab of Tim Yu at Boston Children's Hospital, La Jolla, and other biotech partners. IRF2PPL presented unique challenges. It's a single exon gene containing no introns, and its DNA content is very GC rich, making ASO development especially difficult. Despite these obstacles, we pursued the potential of an ASO treatment until the very end when we determined it was not feasible. Very little was known about Ellie's disease or the disease mechanism, but Dr. Meyer had some preliminary unpublished work and a hunch that gene therapy was our best option to alter the disease course and save Ellie's life. Going off of this hunch and a lot of discussion and prayer, we decided to start manufacturing the gene therapy at the same time as we completed proof of concept and safety studies, an extremely risky and potentially costly decision. But we knew we were running out of time and we had to move quickly. I am now going to pass it along to Dr. Meyer, who will share many more details about the next leg of our journey.
Thank you, Michelle. Yes, so Michelle and Dan approached me to talk about the gene therapy. I actually had started working on this disease in my lab previously because other families also came to me and asked me to do some work. So we had initial data from patient cell lines as well as some initial mouse data using AAV to induce a mouse model that showed some promise in terms of using a gene replacement strategy. So that means bringing back a healthy copy of the gene should probably work. As Michelle already mentioned, so I don't want to belabor the point, here we're looking at a very aggressive, rapid degenerative disorders with a high risk of permanent loss of ability and premature death. We already knew that Ellie was affected. The MRI demonstrated some changes in her brain and the size of her head, so meaning the size of the neurons was also smaller than normal. So we had to move quickly, and therefore I thought that gene therapy, AAV-based gene therapy, would be a really good option because we know very well how to develop these therapies to the clinic. So ultimately, the idea and the initial constructs were already made at Nationwide Children's Hospital, but what also became a little bit complicating is that I was in the middle of changing my career to become the CSO of Alcyone Therapeutics. So we had to navigate those complications as well as I was transitioning to the company and also started my new affiliation with the University of Missouri. This could, potentially delay starting of the program for several months, but we did manage. Again, everybody was working really hard. My friend and colleague, Dr. Smita Saxena and Dr. Andrea Sierra Delgado, picked up the pieces and did execute the safety studies, all at University of Missouri, which also has the National Swine Center that became really handy for the safety studies that we were going to run. We had a very short amount of time. I think Michelle is maybe going to talk about this a little bit also, to find a trusted partner to really execute on clinical manufacturing. Of course, Andolin has been a partner for several of my programs before, so I knew them very well already. And that also helped for me, certainly, to make a recommendation to Michelle. So maybe talking a little bit about the product design. Again, the speed was of utmost emergency here. So the approach that we took was to select only elements that were already being used before, that the FDA has seen. So for the FDA, this should be really a déjà vu approach. Nothing new, nothing complicated, no design elements that haven't been used in the clinic before, so we can predict as best as possible what's going to happen when we move forward and really reduce the risks of novel elements that could create issues or unexpected findings in the clinic. We also used established, previously tested dosing routes for which we know already more or less a window of what's tolerated in the clinic. Of course, there's always risks involved in a first-in-human clinical trial, but again, we do have long-term experience with these elements, with the vector serotype, and then also the manufacturing type that we chose. And then the construct components, we use a promoter that's already been in the clinic, that's been used, that's been published also. We know that it works well both in mice and non-human primates, and that's already published. So as quickly mentioned, in order to move this quickly, we had to really trust our initial proof of concept data that we already had. When Michelle mentioned before, she took a great risk. It was a financial risk. It was not a risk in terms of studies that we ran to get to the clinic, but we did initiate clinical manufacturing and use the clinical material for the safety studies and really move through safety and efficacy studies in parallel, which of course, had we found some major safety issues in our large animal studies, that would have stopped the program at a time point where a lot of the money would have already been spent in order to get to the clinic with this program. But we did run the same amount of studies, number of animals that we would use if we were going in a slower pace and a more consecutive traditional path. In addition to the studies that we ran at Missouri, Alcyon Therapeutics also modeled using MRI images from Ellie's brain the potential delivery mode, and we're able to optimize the delivery mode as well using state-of-the-art novel AI-driven in silico models, so computer modeling, to really make sure that the drug actually gets to the brain where it needs to go. On the left side, you can see, on the right side, you can see images of how that changed the protocol for delivery, and also how it reduced the amount of viral vector that we expect to leak out at the injection site or in the spinal cord. So we are very confident that we were able to optimize this protocol and give Ellie the best chance of success. So Matt, I'm going to hand it over to you.
Great. Thank you, Kat and Michelle. So a little bit about Andelin. We've been an organization working on AAV for multiple decades. We've developed our own proprietary platform, and that's been proven with many GMP clinical batches that supported many INDs. Our one guiding beacon is to accelerate gene therapies and get them to patients as fast as possible. Our vision really is turning hope into reality. We have about a little over 70 programs that are active right now. We are also doing process performance qualifications, so validation runs for clients and commercial manufacturing as well. So we are able to take a gene therapy from initial concept all the way through development, clinical, and commercial manufacturing. Our capabilities include plasmids development and manufacturing, vector manufacturing, fill finish, all the process development and supporting analytics, and getting a program ready for commercial manufacturing. We operate out of several facilities totaling about 225,000 square feet. The platform, and when we'll speak of here for Ellie's program, we term it the Curator platform. It's been curated over many decades, over many programs. It's an adherent platform and a suspension platform, depending upon the cell line selected. Basically, when you start with the gene therapy process, it starts at the bench, right? And then the big question is how to convert that into a manufacturing process. How do you industrialize it? And this is really what the platform, really does. And the way we do that is it's configurable. So every unit operation along the process can be adjusted with levers that we are aware of through many, many productions. And what we do is we implement a optimization strategy, and this is the optimization toolkit that is available to us, and it's backed by a tremendous amount of data. So this process, this methodology for the platform is termed optimization by design, with the ultimate goal to provide consistency and productivity and quality and measure that throughout the entire process. Just a little deeper dive here, look at the platform. So it encompasses several cell lines, adherent and suspension, off-the-shelf helper plasmids. There are plasmids, multiple plasmids that are utilized in the transfection process for AAVs. We have baseline process parameters that have been set and a library of platform assays. And you can see here some of the levers that we can toggle at each unit operation, and we have the platform assays all along the way to ensure that we have the titer necessary and have the quality necessary. So for Ellie's program, we looked at all of the factors, right? And we do this for all the programs. What is the best platform to utilize? Is it adherent? Is it suspension? Ultimately, speed was the main driver. And we need to get a therapy manufactured for Ellie. So looking at the scale and looking at what could we do the fastest, we chose the adherent platform. And you can see on the right here, that includes hyperstacks, and then your basic gene therapy unit operations you're familiar with, clarification, concentration. We leveraged Dixonol ultracentrifugation, column chromatography, more TFF to concentrate, and then final fill to get it into the vial. This platform essentially is ready to go. It's very predictable in terms of yield and quality. And as mentioned, it's been proven over just a tremendous number of processes, several of which have ultimately gone on to a commercial stage. In terms of being able to streamline the quality review and IND, the platform also was ready for this. We have drug master files with the FDA, standardized batch records ready to go, mentioned the platform assays, and all of the release specifications already predefined. So very rapid moving into research production and GMP. So ultimately, with all of the work that Michelle did around learning about the options and learning about gene therapy, getting engaged with all of the appropriate partners, bringing everyone together, Ellie was able to be dosed 14 months following her diagnosis. Just walking through some of the timeline here, if you can imagine this for those in the space to understand the challenges to do this, it's quite amazing and really, a lot of miracles along the way and a lot of teamwork. So plasmid production began only 4 months after a diagnosis at Charles River. cGMP vector production was initiated 3 months later in November of 2024. And then the drug product was tested and released in January. Prior to release, some of the material was released at risk in order to do toxicology studies. This was pig studies, essentially testing for safety. The IND was filed on February 17th, and as a standard, FDA offers a 30-day review. There were 0 CMC issues, and Ellie was approved to be dosed, and she received the therapy on April 3rd, 2025. Just sharing a little bit of the data here. So it's very important that you see comparability between the research and the GMP. And this is the predictability we spoke about. If you look at the process performance from a recovery standpoint, you're going to lose some of the vector along the way as you go through the different unit operations. And you can see here, there's a steady reduction as predicted, but it matched from research to GMP, ultimately landing around the 40-45 percentile recovery, which is really good. There's a list of release specifications. Some important ones here are the purity, which is basically what percentage of full particles versus empty, with the target of over 80%. This was achieved at 86%. Titer, obviously important, making sure that you get the amount of therapy necessary. And we achieved 5E13 viral genomes per mil. Host cell protein, you want to make sure that's very low, target of less than 80, and achieved was less than 1 nanogram per mil. And I will pass it on to Michelle now, who can speak to Ellie's progress.
Yeah, thanks, Matt. So, as Matt indicated, we treated Ellie on April 3rd. The treatment was safely tolerated. Her immune response was stabilized. Based on imaging, we know the treatment was successfully delivered to the brain. And to date, there is no indication of disease progression, and Ellie is developing. As we know, with gene therapy, full efficacy takes time, at least a year, often multiple years. But Ellie is doing really well. We decided after the difficulty of the past 18 months to kind of take the summer off and travel with our family. You can see here a number of pictures of our summer. Ellie's happy, she's smiling, she's at home with her siblings, playing and rolling, and we are incredibly hopeful. We remain steadfast in prayer, and we hope and believe that the treatment has changed the disease course and ultimately saved her life. We've shared some happy pictures here. Obviously the start of the story and the story as a whole is very sad, but ultimately it's also a story of hope, a story of joy. Throughout the past 18 months, despite a lot of the difficulty, we also experienced a lot of really joyful moments, and we really appreciate every single, minute and day that we have with Ellie and with our other children. So beyond Ellie, what's next? There are many times, honestly, that I'm still pretty angry and shocked that this happened to us. But every time I go back to kind of consistently soul searching and praying, and I'm able to replace the frustration and sadness with a deep sense of purpose. I know and believe that we are called to make the world a better place. And very early on, after Ellie's diagnosis, I randomly came across a quote by Margaret Mead. It said, "Never doubt that a small group of thoughtful, committed citizens can change the world. Indeed, it is the only thing that ever has." If you're here today listening to this webcast, and if you're part of this industry, you are really part of this small group changing the world. The science and the development of gene therapy is ultimately changing the disease course for thousands and thousands of kids that desperately need it. Today at Ellie's team, we are also committed to continuing to change the world and to work for treatments for all kids with NETMS. As such, as a foundation, we're continuing to fund grants and research in multiple areas of treatment, and we're planning to continue to expand the gene therapy to other patients. We also remember at the beginning how important and critical it was for us to talk to all of the other rare parents who came along before us with other diseases. They guided us in ways we never could have figured out on our own, and they understood the deep sorrow but intense purpose that we had. As such, we're committed to being a resource for other families afflicted with ultra-rare diseases to try to help everyone get to treatments for their kids faster and cheaper. It's the parents often who are really driving change, and we are so lucky to have incredible organizations like Angelin and Alcyon and partners who are willing to come along with us on this path to push the science, to push the timeline, and to push the costs. So if you're a rare parent and you're interested in connecting, please feel free to reach out to us on our website. We're here to help. Back to you, Matt.
Great. Thank you, Michelle. So in summary, I think it's really important to look at the overall timeline, right? Everything we're talking about here is accelerating. What's that accelerated pathway for a gene therapy? As has been mentioned, it's all about determination and conviction. And once you have that, then it's putting the right plan in place. And this plan included manufacturing at risk, and there was a lot of coordination. So it's a very simplified timeline, but gives a good picture from when Michelle and the family noticed onset of symptoms with Ellie, and then going through all the process to get her an actual diagnosis so that a solution can be potentially found. That was back in February of 2023. I mentioned plasmid manufacturing. You then need to know who you're going to work with for manufacturing. There's a process to get the contracts in place. That takes several months. And then once that's established, we went into a yield assessment, which basically gives you the proof of concept that the process will work. We received the plasmids and began vector manufacturing in September. Material was shipped to University of Missouri for safety and potency testing. We need to also prepare a titer assay. You need to qualify that for anything that's going to go into a patient. So that process was started, and then you go through the process of testing and releasing the GMP batch, and then the IND was filed, approved, and then ultimately the therapy was administered in April of 2025. So this was over a year, but it for those in the space, it can take many years to get a gene therapy approved for the clinic for dosing patients. So it's a really good story on how to move fast, overall for gene therapy programs, but especially for foundations, for families. There is a way. Funding is important, but it can be limited. And if the right strategies are put in place, the right partnerships put in place, and everyone is working together, it can be accomplished. So if you are looking for more information, if you are a foundation, if you are a family, you're not sure what to do, feel free to reach out to any of us here. For Andelin, we have a dedicated business development for nonprofits, so we support foundations, government institutions, academic institutions, anyone that's nonprofit. So we have essentially a toolkit and could really help in the development and manufacturing strategy, cater to specific programs. We like to stay very involved in the rare disease space. We have an annual golf fundraiser. Every year we pick one patient to help fundraise. Ellie was selected in 2024. This year we helped raise some money for Cure CMT4J Foundation for Talia Duff, who's a patient with this disease. And then Ellie's team, Michelle, set up a full foundation. You can go to their website and find a lot more information, and can reach out that way as well. If you scan the QR code, you can get more information about the story. There's a full 4-part blog series that really walks through all of the details. So a really good training ground if you're just getting started. So lastly, a big thank you for joining and for attending and for listening, and a huge thank you to all the contributors that came together that made Ellie's therapy possible. So thank you.
I'm going to pop back on. Thank you so much to Michelle, Matt, and Catherine for sharing that wonderful information, this great story. It's so important. And I just want to say, if you have not submitted a question and you'd like to, you can do so now through that console on your screen. We do have some audience questions that have come in. I'm going to go ahead and bring our speakers back on, and we're going to get to those audience questions. We'll get started with our first question. I'm going to check down here. This question is asking, what was the experience navigating the regulatory landscape for such a condensed timeline? Who can help with this question?
Yeah, thanks. I can take this one. And Kat, please feel free to chime in afterwards. I'd like to say the FDA was incredibly helpful throughout our entire process. We had a pre-IND very early on in the process where we outlined what we were going to do. They were incredibly helpful with that kind of process and guiding us. Ultimately, they agreed with everything that we outlined, and even told us some of the things that we had outlined to do that we didn't actually need to do given the circumstances. They also allowed us to interact directly with them from the foundation perspective and, to email our IND in versus kind of, setting up a portfolio on the online kind of system. They were very accommodating to the fact that we were a small foundation and given the severity of Ellie. We also had kind of open question line with them. We could reach out to them throughout the process, and we did, despite the fact that this was, a very accelerated timeline, and it almost seems like everything went perfectly. It didn't. We had many moments where we actually had hiccups that we had to work through, and questions that we had to figure out. And we often went to the FDA to get their feedback to make sure we were making the right decision. So overall, the FDA was incredibly helpful. As I mentioned, we submitted the IND. We got a number of questions during the 30-day review period, which all made sense. We addressed those in a timely fashion. And then we ultimately got the okay to proceed, exactly 30 days after submitting. But I do really want to just indicate that the FDA was incredibly helpful throughout and has continued to be helpful as we've thought about moving the treatment forward.
Wonderful. Thank you so much, Michelle. Back to our audience questions. And this next question is from Ryan. And Ryan is asking, you noted that your therapy here used AAV9 because it has been previously demonstrated in the clinic. In recent years, there's been tremendous development of novel capsids with enhanced capabilities. What will it take to facilitate clinical adoption of these next generation capsids? Who can help with this one?
Yeah, I think I can start, and Matt, maybe you can add also from a manufacturer standpoint. It's a very good question, and I think it's an exciting research field that is developing very quickly. In a situation like here where we have to move very fast, I would be extremely cautious to go with a new capsid for many reasons, starting with manufacturing. Do we know that we can really deliver on the yields we need? We will probably need larger animal studies with more doses to really understand what is the dose we're going to need. Whereas, with AV9, I was very comfortable based on the research that we had done and based on the data that we already had from previous programs. So in terms of moving quickly, using known elements and the known manufacturing process really, really helps. So what would it take? I think it takes more money, takes a little bit more time and more time spent in the animals to really understand how you're going to move this forward into humans. And it is a little bit of a greater risk for a first in human as well, because ultimately, none of the models we use preclinically are humans. They live in a very defined environment. Mice are often genetically related to each other. So it's just a little bit more of a black box in the clinic. So a little bit more risky from that part, but can be very exciting, of course.
Yes, thanks, Kat. And certainly, more data, clinical data, a better understanding of these capsids is critical. From a manufacturing standpoint, we are seeing more and more clients come to us with novel capsids. We also have some licenses for some novel capsids, the MyWay AVs, and it's a process, right? We bring them in. We need to do a yield assessment. Not all novel capsids are created equally in terms of manufacturing capability. There's great hope for them as they drive added specificity to targets within the body, but we are working on them, and many innovator companies wish to use them. So we bring them through the same rigor as any other established capsid, doing yield assessments, checking quality, and going through the scale-up process.
Okay, thank you both. So we'll hit our next audience question, and this question is asking, I have a question for Michelle. You mentioned that your organization is faith-based. It seems like we often see faith and science pitted against each other, but could you share your thoughts or view on how they worked in tandem for your daughter and in this process?
Yeah,
Michelle?
thank you. Sure. So I can't imagine having gone through something so difficult and challenging and continue to be going through something without my faith. I, work in business outside of what I do for Ellie's team. And, often faith and religion is, external to the business world and external to what you do. But as a mother, when you go through something so dramatic and so difficult, I don't think that you can ignore it. And so it became, essential part of our foundation. It's become an essential part of our journey. I believe that we have been, given a lot of opportunities and paths in order to get something done that otherwise couldn't have been without some divine intervention. We often think of miracles as kind of the big, someone got up from bed and started walking. I actually believe with my faith that the miracles are happening every single day for Ellie and for us. And that became very clear throughout the 14 months. I continue to stay kind of steadfast in that focus. And I found, many partners in the science world, many that are, listed on our team website and here that believed that with me and came along with that with me. And I think a lot of the team, if you're very, very involved in our journey, saw the daily miracles. It was honestly very hard to ignore. And while we put our faith in the science is still limited. There are so many things that we don't know and that we'll never know. And so ultimately, my husband and I, and our family and many of our team, while we believe in the science and we push in the science, we ultimately have to believe in something bigger.
Thanks, Michelle. Okay, next question is asking, could you please elaborate more on the imaging technology procedure to track the delivery of the therapy? Who could help with this one?
Yeah, I guess that's for me. So the imaging itself wasn't something novel or complicated. We used imaging reagents that are commonly used for imaging in radiology. But what was really unique here is the modeling that we used to optimize the delivery technology, which is unique and proprietary to Alcyone Therapeutics, but lucky enough, we were able to use it and work with Ellie's team and get that done for this case. And it is really based on understanding the CSF flow, so the spinal fluid where you put the drug in. It doesn't just flow around in a steady state. It's pulsating. There are hindrances, there are nerve rootlets that are getting in the way of moving your drug. And so we are modeling these things and really trying to understand how can we get the optimal biodistribution for the disease that we are trying to treat.
Okay, very good. Thank you so much. So the next question is asking, do you anticipate that the patient will need to be retreated at a later time and point? Who would like to take this one?
Yeah, maybe I can start with this, Michelle. So we know that AAV treatments are very long-term based on the data that we have, especially in the nervous system, because the neurons don't divide over time. So once you treat the neurons, they should be surviving as long as you are able to really stop or change the disease course in the cells. But nobody knows if it's going to be forever. It is very possible that in 10 years or 15 years, a second dose might become necessary. And there's a lot of people that are working on redosing protocols, which is not that simple because once you give the therapy to the body obviously develops an immunity against it. This is seen as a viral infection to the body, and so you will have to combat that immune reaction for a second dose.
Yeah, and maybe just to add on to that, I mean, we know that the treatment that was provided to Ellie was not a cure, and it was not the absolute perfect treatment, but it was the perfect treatment to us that was available at the time with where the science is at. So Ellie's team is continuing, as I mentioned, to fund grants and to fund research and to keep our pulse on kind of all of the evolving technology. We are, focused on gene editing. When ultimately gene editing is figured out on how to deliver it to the brain, we hope to be ready. So, there are many other aspects that we are continuing to focus on. But we do believe that we made the right decision and we took what is the best science available to us today. And we did that. And we do think that it is going to be a meaningful treatment that we were able to accomplish.
Thank you, Catherine and Michelle. Heading back to the audience questions, this next question is asking, the team mentioned that there was a direct focus on making sure the treatment was delivered to the brain at a higher concentration. Could they elaborate on this?
Yes. So when you use the CSF delivery for the biodistribution of AAV9 or any AAV or any drug really, it doesn't just evenly distribute the way you want, right? And it doesn't matter if you use a intracerebral ventricular injection to get to the spinal fluid or a cisterna injection, which is at this level, or a lumbar intrathecal injection. The biodistribution for any of these methods is uneven. So you have higher concentration in certain areas, and the drug doesn't always flow in the direction that you want it to flow. So sometimes when you do a cisterna injection, which is also what we found with the baseline protocol, a lot of the drug would actually have leaked into the spinal cord instead of getting to the brain. And because it's not a closed system, there are leakage pathways out of the spinal fluid space. That drug will never make it to the brain. It might go to the periphery. It might end up in the liver, which can be a problem because it can create side effects. And so the protocol that we optimized really made sure to the very best we could to get as much drug to the brain as possible and distribute it as evenly as possible so that all of the brain is basically soaked and exposed to the treatment. There's still areas, of course, that are very difficult to reach, which are the deeper brain structures that generally get less of the vector. But again, by avoiding the leakage, we already have a higher chance to get more to the places where it needs to go.
Okay, very good. Thank you so much for that. Now, this question is asking, was this a single patient IND? Was it a manufacturer for a single patient? Can someone help with this
So
one?
we applied for it. Yeah, I can take this. So we manufactured the smallest batch possible at Angelin, just given cost concerns. This entire program was kind of self-funded by Ellie's team. There was no institutional money or investors or anything like that. So we had to try to keep the costs as tight as we possibly could. So we manufactured the smallest batch possible. The IND that we submitted was for an N of 1 to treat Ellie. It was not an emergency use IND, though. It was, a regular research, issued IND. And the FDA has indicated support to expand the IND to treat other patients.
Okay, very good. Thank you so much. Now, this question would like to know, did you have to generate multiple tox and GMP lots before the final GMP product was administered to Ellie?
Yeah, no, so
No,
that's...
we produced...
Sorry.
Oh, go ahead. Okay,
I was just going to say that's... go ahead.
Okay. As mentioned, cost was, top concern. And so we produced one research grade batch and one clinical batch. And the research grade batch was used for our tox study in mice and for our proof of concept work. And then a small amount of the clinical batch was used for our safety study in the pigs.
Yeah, and I was going to say that is the beauty of, this accelerated approach. Really, technically, you could run all your studies with the clinical batch. And that way you don't have to worry about comparison between the batch manufactured, release testing, etc., but also the concentration. So it gives you additional confidence, in particular with the large animals that you use to scale your dose, that your safety data really is going to be applicable to the patient.
Okay, thank you. Isabel would like to know, what strategy do you anticipate to bring forward to generalize this approach for more patients? What do you think?
So is this for more patients of the same disease or for more patients of different diseases? So depending, I can say for the same disease, of course, the manufacturing we used, as Matt pointed out, is established. So we can make more viral vector, we can expand the IND, even hopefully move, to other countries and start treating patients in other countries as well. Of course, all of that requires resources. And we require, additional funds for Ellie's team to continue moving this forward. But as Michelle said, the foundation is very dedicated to move and develop this drug for more patients as well. Matt, did you want to add anything in terms of the manufacturing platform and how it applies to other diseases as well?
Sure. So, what we do is we look at every program and all of the different factors and determine which is the best platform. And so for this one, we selected the adherent platform, and that can be leveraged for specific programs depending on the number of patients. If there are a wider population required for a specific program, then we could look at a different platform. So you really cater the program needs to the platform.
Yeah, I think just one final thing to add on this. Obviously, when I talk to other parents who are trying to kind of move forward programs for their children, there's just a lot of question about what does the FDA require. I think a lot of people are doing really amazing work in this space to try to kind of share what the FDA required for their program or for our program. And while every single disease is different, having kind of these open interactive sessions with the FDA in order to try to move things forward faster and quicker. And as I mentioned, the FDA was really phenomenal with us. And so, I want to give them kudos for that. And I think, interacting with them and really just trying to see what is it do you really need to do, which can help limit costs and accelerate timelines.
Very good. Thank you. Now, this question is asking, how many of N of 1 indications can be manufactured at Andelin so that this process becomes more streamlined for more families with children with ultra-rare genetic diseases? Matt?
Sure, thank you. Yeah, ultimately, we've been able to address any N of 1 program that has come to us. We have a fair amount of capacity in our commercial facility. Much of it is populated by biotech programs, but we do allocate and keep some capacity available for these N of 1 programs, and we've done a number of them now.
Very good. Well, that is our time for today. I wanted to leave everyone with just a minute to leave any final thoughts you'd like to leave with all of us for today before we end. Matt, Michelle, and Catherine, any takeaways that you'd like everyone to leave with today from our webinar?
Yeah, I just want to say, I'll go first, and Michelle, I think everybody wants to hear from you to close out. But just, again, thanks for the attention. Thanks for the questions. The entire industry and families newly dealing with these situations are learning best practices. So Thanks for tuning in, and hopefully there's been some good lessons learned here. And we are open and available, as are all the partners, to assist in any way that we can.
Yeah, thanks, Matt. I just want to, thank everyone for the time. Thank you for the thoughtful questions. As I mentioned, we're all here to help, so please feel free to reach out to us. I also just want to reiterate how incredible of a partner Angelin was throughout our process. There was a question we didn't get to about how did we make the decision on who to manufacture with. Angelin from the start became like family to us, and they went really above and beyond to get this done. As did all of our partners. And so we could have never accomplished this without, every single one of the people that were part of our team. So I want to continue to kind of thank our team for getting this done, reiterate that we are all committed to kind of moving forward the industry for all the kids that desperately need it, and thank everyone that is here and that is listening and that is working, on treatments and advancing the science for our children, because we really need it and we really appreciate you, and we couldn't do it without the team.
Very good. Catherine, any final thoughts?
Yeah, I think,
Michelle said it all.
Yeah, maybe just some words of encouragement for the families. I can't even imagine, what the journey is for many of these rare disease parents, but I would say these are some of the strongest individuals that I've ever met. And sometimes maybe there is a little bit of hesitance or fear also to reach out to researchers or reach out to, manufacturers or anyone in the space or even to the FDA. But in my experience, I think all of them can be really good partners, and many of them are highly motivated to help the rare disease community. And that's something for me that I've experienced, and I'm also grateful for, to see that even companies can step up and people are willing to take calls after hours to make something happen for these families. So just don't give up hope, I would say. I know it's tough, but I think many people are there to really help and connect and bring you forward.
Very good. Well, I appreciate all of you for your time and for sharing your experiences and your thoughts today. And thank you to the audience for watching. Just a reminder, this webinar will be available on demand. You'll receive a link via email as soon as it's available. Thanks so much. Have a wonderful day. We'll see you next time. Goodbye.